Microencapsulation of aliso, chilca, walnut, and talla dried by atomization. We have introduction, materials and methods, results, discussion, and conclusions. Introduction. Microencapsulation is a technique that allows the packing of materials such as oils, bacteria, enzymes, whey, vegetable dyes, minerals, vitamins, and food additives. The main encapsulants are polyvinyl alcohol, alginates, lipids, carbohydrates, gums, and proteins. The encapsulation can be by physical, chemical, or mechanical process, being the spray drying technique the most important one. The powdered colorants of alder, chilca, walnut, and talla obtained by atomization deteriorate rapidly when exposed to the environment because they absorb moisture and low yields in the spray drying, since they adhere to the walls of the atomizer. Therefore, the microencapsulation of the dyes improves the performance during drying and avoids the deterioration by absorption of humidity, prolonging the conservation time. The objectives of the present work are to obtain microencapsulated dyes of alder, chilca, walnut, and talla, dried by atomization, to determine the appropriate proportions and to check the properties of microencapsulated dyes and their stability. The alder, chilca, walnut, and talla colorants were extracted and concentrated by boiling, microencapsulated with maltodextrin and different concentrations during the drying by atomization. Then, stability was evaluated with humidity and white light exposure tests, and the color and morphology content were determined with the support of Globe Natural Company and the National University of Engineers respectively. Materials and methods: harvesting, cleaning, and drying the plants. The alder, chilca, walnut, and talla plants were collected in the town of Quinoa in February. They were washed to remove soil and other impurities. The selected leaves and tender stems were drained and dried in the environment. Dye extraction and concentration. The extraction was carried out in an aqueous medium, boiling for 45 minutes with a raw material solvent ratio of 1:20. In the case of alder and walnut, it was in 2 stages, due to the fact that 30% of dry tender stems were used. It was concentrated by evaporation until a volume of 500 milliliters, and the adequate percentage of soluble solids, between 5 and 12%, was obtained. Microencapsulation and spray drying. The extract was separated into 4 equal portions for drying without microencapsulant and the other 3 with maltodextrin, in proportions of 20, 30, and 40% for alder, 20, 40, and 60% for chilca and walnut, and 5, 10, and 15% in the case of talla, with respect to soluble solids content. It was dried by atomization at 240 degrees, 4.0 kilograms per centimeter square, with the atomizer Niro Atomizer, Copenhagen, Denmark, number 1592 of the Mass Transference Laboratory. Stability of microcapsulated dyes. Moisture determination. A. After spray drying, it was performed by means of a Hacke humidity determinator from the FIQM Mass Laboratory. B. During 30 days, the colorants were exposed to the environment in wet months for 30 days. The humidity absorbed in different times was determined, taking into account the environmental temperature and relative humidity. Color stability of microencapsulated dyes in white light. It was determined the wavelengths of maximum absorption of each colorant through a scan in the UV-VIS thermoscience spectrophotometer Genesis 6 model of the FIQM research laboratory with aqueous solution at 0.05% of dilutions, alder and talla 1.5, chilca and walnut 1.10% of microencapsulant. Exposure to white light was made in clock moons during 0, 24, 48, and 22 hours. In absorbance readings at maximum absorption, wavelengths were made by ultraviolet spectroscopy. Physical characteristics of microencapsulated colorants. pH. The pH of the aqueous solution of the microencapsulated colorants were determined with the Janeway portable pH meter from the Instrumental Analysis Laboratory. Scanning electronic microscopy. It was carried out in the Electronic Microscopy Laboratory of the Science Faculty of the National University of Engineering with the Philips 500 scanning electron microscope. Color measurement. It was carried out by means of colorimetry in the laboratory of the Globe Natural Company. Of the selected colorants of alder with 0% and 30%, chilca and walnut 0% and 40%, and talla 0% and 10% of maltodextrin. Results. The plants under study were collected in the town of Quinoa, located 34 kilometers northeast of the city of Ayacucho, 3,400 meters above the sea level. The microencapsulated dyes were obtained by spray drying. The moisture content of the various immediately atomized samples and the drying performance are shown in Table 1, and the ratio of moisture content to microencapsulant content is shown in Figure 2. Table 2. Colorants obtained from spray drying and percentage of humidity. Figure 3. Variation of the humidity of the colorants versus the percentage of microencapsulant, maltodextrin. Moisture determination for 30 days. Figure 3 shows the variations of humidity with respect to the time of exposure to the environment of the microencapsulated colorant. During the exposure to the environment, changes in physical characteristics were observed. In the case of alder, at 7 days, the lumpy, caramel, and dark aspect was noticed, being more accentuated in the 0 and 20% and in less proportion in the 30 and 40%. In case of chilca, at 12 days, a slight change was noticed in the aspect of the colorant becoming lumpy, caramel, and dark in the 0 and 20% and very little in the 40 and 60%. These characteristics gradually increased until the end of the trial. In the case of walnut, from the 5th day, a slight change in the aspect of the colorant was noted, becoming more noticeable after 15 days of exposure to the environment, presenting a lumpy, caramelized, and dark aspect, being more accentuated in the 0 and 20% and in a smaller proportion in the 40 and 60%. In the case of talla, after 5 days, it was observed a slight darkening and formation of some lumps in the 0 and 5%, and maintaining the color and aspect in the 10 and 50%, gradually increasing these characteristics until day 14, where it was observed that the 0 and 5% were brown, caramel, and lumpy, the 10 and 15% in less proportion. White light stability of microencapsulated dyes. Determination of the wavelength of maximum absorption. absorbances of microencapsulated dyes exposed to white light. The corrected absorbances of microencapsulated dyes are reported in the Table 2. The variation of the absorbances with respect to the time of exposure to white light, from 0 to 72 hours, are shown in Figure 3. Physical characteristics on microencapsulated dyes. In water solution, the pH was alder 6.9, Chilca 6.8, walnut 6.8, and Taya 6.8. Figure 4. Variation in absorbance of colorants exposed to white light. Figure 5. Scanning electron microscopy of alder with 0 and 30% maltodextrin. Figure 6. Scanning electron microscopy of Chilca with 0 and 40% maltodextrin. Figure 7. Scanning electron microscopy of walnut tree with 40% maltodextrin. Figure 8. Scanning electron microscopy of Taya with 0 and 10% maltodextrin. Figure 9. Colors of microencapsulated dyes are A. Alder 30%, B. Chilca 40%, C. Walnut 40%, and D. Taya 10% maltodextrin. Discussion. Extraction and concentration of colorants. In the extraction of the colorants of alder and walnut, 30% of crushed dry tender stems and 70% of ground dry leaves were used in 2 stages, improving the percentage of soluble solids from 7.12% to 8.54% for alder, and from 4.99% to 10.87% for walnut. Microencapsulation and spray drying. The maltodextrin percentages for each plant were defined according to the characteristics and behavior during spray drying. For alder, 20, 30, and 40% regarding the colorant content; for Chilca and walnut, 20, 40, and 60%, and for Taya, 5, 10, and 15%. In the Table 2 and Figure 3, it is observed that the use of maltodextrin as microencapsulant favors the obtaining of powdered colorants with lower percentage of humidity and better performance in the drying. The lowest percentage of humidity was obtained using maltodextrin in the following proportions: for alder 30%, for Chilca and walnut 60%, and for Taya 10%, and the drying yield improved from 35.83% to 46.38% for alder, from 80.77% to 94.58% for Chilca, from 77.1% to 80.4% for walnut, and from 75% to 82.5% for Taya. Determination of the humidity of microencapsulated colorants during 30 days. In Figure 4, taking into account the percentage of acquired humidity and the changes in the physical characteristics of microencapsulated colorants, it is suggested that the best proportion of colorant maltodextrin for alder is 30%, for Chilca 40%, for walnut 60%, and for Taya 10%. Stability to white light of microencapsulated colorants. The microencapsulated alder colorants that present greater stability to the action of white light are those that contain 30 and 40% maltodextrin during the 72 hours of exposure, so it is recommended to use 30% maltodextrin. The microencapsulated colorants of Chilca that present greater stability to the action of white light are those that contain 60% maltodextrin, but it decays after 48 hours, while the 40% tends to stabilize after 48 hours. The microencapsulated walnut colorants that present greater stability to the action of white light are those containing 60% maltodextrin, but it declines after 48 hours, while that of 40% after 48 hours ascends, equals that of 60% and tends to stabilize. That of 20% has the same initial tendency as that of 40%, but after 48 hours begins to decay. So it is recommended to use 40% maltodextrin. The microencapsulated dyes of Taya that present greater stability in the action of white light are those that contains 10% maltodextrin during the 72 hours of exposure. These behaviors are observed in the data of Figure 7. Conclusions. Microencapsulated dyes were obtained from alder, chilca, walnut, and taya, dried by atomization. The microencapsulated used was maltodextrin because it was more commercial. The maltodextrin colorant proportions used were, for alder, 0, 20, 30, and 40%; for chilca and walnut, 0, 20, 40, and 60%; and for taya, 0, 5, 10, and 15%, being the optimal ones according to the humidity absorption and stability to white light tests: alder 30%, chilca and walnut 40%, and taya 10% of microencapsulant maltodextrin. The microencapsulated colorants dried by atomization present the following physical characteristics: alder, fine yellow powder, soluble in water, pH in aqueous solution 6.9; chilca, fine yellow powder, soluble in water, pH in aqueous solution 6.8; walnut, fine brown powder, soluble in water, pH in aqueous solution 6.8; and taya, fine yellow powder, soluble in water, pH in aqueous solution 6.8. The use of maltodextrin as a microencapsulant favors the obtaining of microencapsulated colorants with a lower percentage of humidity. Thank you.